Welcome to the queenon7.com. We will learn about amino acids. My previous lecture was about the amines. Today we will look at the amino acids very quickly. Any carbon atom, when it is attached with the COOH group, carboxylic group, and amine or group on the other side, that will be known as amino acids. To name them, I've got some examples there. The simplest one is glycine, but according to IUPAC system, where you actually use the rule to name the side chains and the longest carbon chain, you look at this example, you have 1, 2, 3, 4 carbon atoms there. The priority will go to the carboxylic group, and the amine group will be known as the side chain. So you will look at it, it's a 2 amino, which is there, amino group. You start the counting from there, 1, 2, 3, 4 on the top. So the position of the amino group is a 2 amino, then 1, 2, 3, 4 butane, then oic acid. So 2 amino butane oic acid will be the name. Then the second one is, which is the simplest one, you count the number of carbon chain, 1, 2, the priority will start from there, 1, and then this will be 2, so it will be 2 amino, because it came from the ethane, the parent hydrocarbon, so it will be ethanoic acid. We will look at the properties of the amino acid. Amino acid does not actually present in the form of the neutral molecule. They actually they are actually present in the form of the ion. And if you look at the structure, this is actually the structure of the amino acid, a general simplest amino acid you have. This R group can be anything. It can be a carbon chain connected with the carboxylic group or amino group. This is called Zwitter ion. No amino acid is actually present in the form of neutral molecule. They all are present in the form of the Zwitter ion, where carboxylic group will lose the proton and the amine group will actually gain that hydrogen. One thing I want to mention here, which is pretty unusual about hydrogen bonding. If you look at it, the hydrogen bonding is the bonding between hydrogen and oxygen, fluorine, and nitrogen. In the neutral molecule, you can have the hydrogen bonding, but in the ionic form, which is a Zwitter ion, that will oppose that hydrogen bonding and will take over, and it will be present as the ionic interaction of the molecules with the other molecule. I will not get into the deep chemistry of Zwitter ion, but I will do explain the Zwitter ion behavior in different sort of solution. So if this is the Zwitter ion and you will actually add an acid, or you treat it with any alkali, so any source of alkali or any source of what do you call it as an acid, will give you two different results. This hydrogen will actually combine with the carboxylic group, so that NH3 will stay as it is, which is positive charge, because nitrogen has got 4 bonds in it, so it will carry positive charge. This R will stay as it is, and this COOH will be converted into carboxylic group. Whereas on the other side, if you are actually treating amino acids in an alkaline condition, you will see that the NH3 will be converted into NH2 with the carboxylic group staying as carboxylate ion on that side. Obviously, this OH ion will remove one of the hydrogen from the NH3 group and will convert it into NH2 group. If you look at the esterification, that's the next reaction we're going to look at. Esterification of amino acids are actually pretty simple if you look at it. If you do remember, in GCSE you have done the chemical reaction between the carboxylic acid and alcohols. Alcohols and carboxylic acid, they undergo the condensation reaction and they will produce a condensed polymer along with the water molecule. It will go same as for these as well when you do esterification. If you do have an amine which is a simple amine, and you're going to react with any alcohol. You can pick any alcohol, and you probably do know that the carboxylic group is actually an acidic group, but it will not release proton in this reaction. It will undergo the condensation reaction, and the OH will come out, rather H positive ion. Now the water will come out, and the product will be an ester linkage, which will be written as this. So this is called the esterification, and you will also produce water as a product. Now, I did tell in my lecture in the carboxylic acid, where I emphasize whenever you do the hydrolysis of the carboxylic acids, or the derivatives of carboxylic acids, like acyl chloride, you have esters. All of these, when they undergo the hydrolysis, they will always produce their parent carboxylic acid. So one of the product will be actually the carboxylic acid. So if wherever you can see these sort of bonding there, so this part will be broken down, and it will be converted into NH2 R bond. Sorry, this one is hydrogen there, and then this is hydrolysis. And when you actually do the hydrolysis, the rest of the part will be the alcohol which is produced after this reaction. Okay, so that's the esterification of the amino acids. Now, if I want to do further esterification, I want to involve NH2 into this chemical reaction. All I have to do is I am just going to remove this. If you want to take a screenshot, you can take a screenshot. Otherwise, I'm going to remove it.